Hi, I'm Emma, and I'll be presenting our project for the Computational Genomics course. Our project was to implement variant calling algorithm based on binomial distribution. To summarize, implementation was done in Python, our input was in pylab format and output in VCF 4.2 format. For testing, we used these BAM and reference genome VESTA files. And in the end, we also compared results with BFC tools call. To gain better understanding of the algorithm, here is some introduction. Variant calling is the process of finding differences between reference genome and observed sample. There are many different types of genomic variants. Some are single nucleotide variants, insertions, deletions, but also larger ones like translocation or whole genome duplication. Some can be harmless, like longer eyelashes, but others can cause cancer, so we need to keep a track of them. Here we can see the format of a pylab file. It has sequence or chromosome name, position, based on the reference genome on that position, number of reads matching that position, and read results and quality. Since reads are matched both in forward and reverse strand, signs for base matches and mismatches are a bit different. So here we have like dot and comma, both mean the base is matching the reference base. And also here is VCF file format. Some information is the same as in pylab, like chromosome position, reference genome, but we also have information about genotype and alternate alleles and so on.
Hi, I'm Nicola, and I will be presenting the second half of our presentation. We will need the binomial distribution to choose variants. Binomial distribution models the number of successes in a set of yes and no trials with equal yes probability p. In our case, trials are nucleotids at one position, yes is a correct nucleotid given variant assumptions, no is a read error, and p is a fixed probability of reading a single nucleotid correctly. Human genome is diploidic, hence at most two variants can be at one position. We will discard all variants except the two most common ones. Let's call them A and B. Let's assume there are k A letters and n minus k B letters in the pylab line. We will choose the variant for which the letter counts are most likely compared to other variants. If the variant is AA, k nucleotids are correct. If the variant is BB, n minus k nucleotids are correct. If we have diploidy, both are correct. In this case, we will assume that A and B should appear with same probability in order to calculate the likelihood correctly. We will just pick the most likely one. After we are done, we store the results in VCF 4.2 format. We will compare our file with the referent one to see how many variants we got correctly. The referent file gives us true labels, either has variant at position or doesn't. Our file gives us predicted labels, and we calculate the precision, recall, and F1 score accordingly. Here are the results we got. To test the algorithm, we ran it for probabilities between 0.5 and 1. And we can see that the recall goes up with probability p, while the precision stays the same for almost all probabilities except for really high ones. Here is the confusion matrix for p equals 0.8. That's the probability that gave us the best F1 score. Possible improvements to our algorithm are to use a different probability for different pileup lines, and to use read qualities to calculate the likelihood of one nucleotide being correct. That's it. Any questions?